I'm showing a screen that's showing this article, but the whole point was to just find some more research on the co-culturing of fungi, which it seems has been going on for some years, a few years, but it's all behind paywalls. So people have not like caught on to the hype of this and this technique. And perhaps it's maybe not justified in the commercial level, which is usually everyone's entry point into mushroom cultivation. It's like always the commercial level or like for your food. But once you start getting into like this scale of like biochemical testing and analysis and those type of therapeutics, you kind of think in a way, and it seems like a motif, you start to think in a way that you're like, all right, I don't need to go all the way down to the fruiting body. I just need to see what these species are exerting at different times. But some of the research I see that as a trend as well right now. But some of the research I want to do is prove that that is, or at least understand if that's like practical or smart, because at different stages in the cultivation process, we introduce different substrates and ingredients and nutrients. And so those would also serve as activators for certain gene clusters, which may be silent otherwise. When it comes to science, you can never just do one thing. You have to do a series of assays that creates this overwhelming compounding amount of evidence that through those experiments and its linked knowledge and references behind it, establish the lore, establish the story that's told about whatever you're researching. Usually whatever you read, it'll be consistent as far as how they're testing this kind of stuff. So we're going to see the same assays. We're going to see hopefully similar or not refuting evidence of this increase in bioactive compounds through the co-culturing. Oh, look at this non-competitive co-culture. This is something we haven't seen yet. So most of the justification for the co-culturing has been that the antimicrobial agents or compounds that are released are in due part due to the competition, the predation of another species, because that other species is going to be releasing their compounds to either protect themselves and whatnot. But that's only one dynamic of interactions. It's not always territorial or problematic for two species interacting. It can be mutualistic. And so here we have evidence of a non-competitive co-culture. It seems they can, which is what we actually want to find more of as a more non-competitive relationship, because that could probably scale and integrate with commercial levels. Because when we start to get to competitive interactions, that will almost always limit the output, right? The final output. So if we're competing, it may or may not result in a lot of what you want. It may result in limited amounts. But here we have a non-competitive co-culture, which is what you probably see with like a mycorrhizal species, right? When we have the cyclic dynamic, we'll have bacteria and fungal species with the plants interacting. And that doesn't seem problematic, you know what I mean? And so, or competitive in that sense. It seems very mutualistic, and it seems like it's helping each other. So maybe it's alleviating metabolic stress by divvying up the roles and allowing each other species to be tapped into their own resources and their own communications, and then they're able to come together. So if we have a non-competitive co-culture, that is kind of what we're probably looking for, specifically in mushroom cultivation. Yeah, we want antimicrobial compounds. We want antibacterial compounds, antifungal compounds, and you'd probably co-culture. It seems logical to co-culture whatever you want to produce it to be that species of anti-agent to be produced. Does that make sense? So like if you're using fungi and you want that fungi to produce antibacterials, you'd probably introduce a bacteria to it. Versus antifungal agents, you want more of that, you'd introduce maybe another fungal species in your broth or on your plate, whatever stage we're going to be testing and doing this. Now, one of the papers that I saw mentioned that this was in the abstract too. I'm not giving any like paid stuff away, but it talked about how it's bidirectional. It's bidirectional, the activation of these silent gene clusters and the compounds that come. So it's increased production of compounds and potent compounds, but it's also novel compounds being constituted based on this bidirectional activation. And that isn't always competitive. Here we're seeing non-competitive. I think this is just a good link, y'all. I think this is really the next wave, the next thing that's going to be added to the cultivation process, because right now it's just like a rinse and repeat method. You know what I mean? Take a tissue sample, put it on a plate, transfer to LC, LC to grain, grain to substrate, fruiting body, extract, standard process. But a lot of this research basically proves that puts that final product that you have is like sealed. It's in a range of medicinal potency that we can test. All right. And people aren't probably finding ridiculous, crazy strains that have that anomaly, like penicillin anomaly. It just produces crazy amounts unless they're GMOing it. But here, when we introduce environments and relationships and dynamics that are seen in nature, we're going to be able to tap into those new ceilings of potencies of novel compounds, right? If we compare those, what do you find in nature? What do you find in vitro in the lab? In nature, you're tapped into a lot of different resources. You can communicate with different organisms and be in a competitive environment. And so you're activating silent gene clusters. But in vitro, you're tapped into your container. That's all you have. And so by activating more things, and it's no longer being a monoculture or axenic, as a word that I saw recently, we can activate those same silent gene clusters. And the research proves that. And this has been going on, but it's behind paywalls. So yeah, if you know anybody that's highlighting this kind of stuff or is doing this practice, like we gotta highlight that. We gotta build it up. So, because this is definitely going to be the new standard for sure. Like there's no doubt about it. And there's further implications, right? I'm currently not seeing these researchers compare. So they're often stopping at the LC stage, right? They'll go plate, monoculture or isolates and mix, right? They'll mix them in the LC stage or their broth stage. Filter, go through that process. They incubate them, let them like cook for so many weeks, produce their metabolites and whatnot. But in our typical production, we go all the way down the line, and I don't see them testing or comparing this same culturing at different stages of development, right? Because at different stages of development, we're introducing different ingredients, different concentrations, and like different substrates, right? And so that should also serve as activators for certain gene clusters, just like competition is, or another species in a non-competitive way is activating silent gene clusters. So I'm not seeing them do that research. I'm not seeing them compare it, I should say. And so there needs to be a standard first off before we just choose to do all the research at the LC stage. And that's what seems to be happening. The co-culturing is only going to the LC stage, and then the analysis, filtering, all that kind of stuff gets done. Meanwhile, in the actual practice in industry, we are not stopping at LC. You know what I mean? Same thing with like just serving the myceliated grain, the fermented grain. Paul Stamets gets so much crap for that. So much crap for that. Like, it's just rice. It's just rice you're eating, even though it's myceliated. And there's testing that's proven that and it builds on what we're saying. Different substrates, different interactions, different exposures activate different clusters, gene clusters, different regions of the genome that code for proteins that produce other compounds. And so there's research out, and typical research is contained in this one stage of liquid culture. So I think it's good. I think we need to see that because the implications, right? Why are they even doing it there? Why are they only stopping there? I mean, it's kind of obvious, but it's not because, right? Because industry doesn't do that. Or as far as like commercial scale doesn't do that. But the implications are that the time, right? How long it takes to produce the fruiting body all the way down there and then process it from there. Right? There's so much more risk involved with contamination or uncontrolled contamination. And right, so in a way, it's like they're trying to identify a space to introduce this into the industry, into the normal standard practice, and they're trying to justify it through a commercial business perspective, but it's not matching what is done in practice already, because like people will still need to produce the food, that final product, you know what I mean? So, and again, they're not testing like, hey, what are the concentrations of these novel compounds later on? Are we still seeing it later on? Can we do this on a block substrate, this co-inoculation? But yeah, we're going to highlight this one because of the non-competitive co-culturing, right? So if we look up some of these other ones that I was pointing out, again, those are competitive, right? So it's like, I'm going to introduce another fungi, and that will help me produce antifungal agents, or I'm going to introduce a bacteria to produce antibacterial compounds more in my metabolites. You know what I mean? And so, but that again, that's competitive. That's territorial, problematic in a sense. And for me, what I think when I see that, I think that that's going to be limiting the yield, right? So that's going to have problems with scaling up into and integrating into the standard commercial level because of yield. If you're in a competitive state, that's it's probably going to lead to, okay, I'm not logged in. Yeah, it's probably going to lead to limited yield. I would imagine. I can't prove that, but this up. All right, y'all can see that, right? So we have non-competitive co-culture. A lot of this research shows like species I'm not commonly working with, but I have found some that talks about like Pleurotus austratus, your oyster mushroom, Trametes versicolor, turkey tail, and it makes sense. Like our Ganoderma species as well. It makes sense. Those are your more medicinal potent strains that people are using specifically for the medicinal biochemical compounds. So I think that's why those are typically there. But yeah, I've seen some of the more commercially commonly cultivated ones being talked about in some of this research. And even if that's limited, I think that's good because that leaves room for all of us because, right, we have those cultures. We're already doing that practice. Now we can just start right away. We can start co-culturing. You know what I mean? If you, whoever's watching, maybe you got some plates right now and you're going to go start co-culturing. I just poured plates yesterday. And so I'm leaving them. I usually leave them for like 3 days to QC, but yeah, I'm doing my breeding project for the new wild strains, and I think I will try some co-culturing. I don't know. I'm excited. But all right, let's get to this abstract. So we got microbial. So yeah, I can show you guys the abstract because everyone has access to the abstract. But it's not until, you have to get to the good stuff, you have to be logged in with a paid account or access through your institution, which is probably a good idea. Like y'all should get this stuff if you want to stay on top of your field, because it's really expensive to get a pay for every article. So microbial co-cultivation is a strategy to induce specialized metabolites, right? This is more hyper-focused. So when we think about the typical practices done, right, it's typically through genetic modification. You know what I mean? Overexpression. We're going to put a known promoter and we're going to introduce, various genes and activate processes, regulatory metabolic processes through that means. But here we can do it through what's the word? Proximal, local proximity of other species, and they're going to do what they're going to do. You know what I mean? It's nature. That's what they do. So most previous studies focused between competitive interactions. You see that? Between test strains. Okay. Basidiomycetes fungi. I've never heard it put like that, but that's what we want to look into, Basidiomycetes. Chemical screenings. Okay, we discovered that the co-culture of Felinus orientoasiaticus, I'm so sorry I butchered that, y'all, Hymenocetaceae, I'm so bad at this. This is so bad. Xylodon flavoporus, Schizoporus. This is so bad. I should stop. Induces multiple metabolites, although they did not show any competitive morphology. What would be competitive morphology? Competitive morphology. Are we talking like visible? Like if it's on a plate, like we see the zone of inhibition, would that be a competitive morphology? Or am I just way off? I could be totally way off. So we have targeted isolation yielded 3 sesquiterpenes along with 5 known analogs. Oh my goodness. This is so another crazy reason why this is so powerful, because even if you're not getting novel compounds, we could be finding novel biosynthetic pathways to get to the synthesis of certain molecules. You know what I mean? And if we can get to analogs through various co-cultivation processes, that eliminates synthetic chemistry, that eliminates synthetic biology, or at least it alleviates some of that, like that mindset, that motif. What's the word? Dang, I'm blanking on my words today. My zeitgeist, the zeitgeist. I feel like it's a zeitgeist, right? To just go for the genes and just do gene therapy. I feel like we're in that, rather than identify the pathways a little more differently and find different ways to manipulate it. But yeah, having this as a technique of varying the co-culturing to activate different biosynthetic pathways to produce certain compounds or analogs of those compounds, that can accelerate the structure-activity relationship mapping for these medicinal compounds. If you haven't seen my mycochemistry video, go watch that as well. And yeah, this is crazy. This is actually kind of nuts. This is so powerful. This is so powerful. And that's scalable. It's scalable. Oh my goodness. If it's just co-culture. And again, we need to see them compare what they're doing, these co-culturing, extraction and analysis breakdowns at different stages of growth. We can't just do this only at LC, at the liquid 3D stage. One, because of different substrates, right? In the wild, They're going to be on like hardwoods, right? Where's the hardwood in your liquid culture? They're not adding it in there. You know what I mean? There's room for so much improvement, but we're finding the thread to connect or to give guidance on where we should push the industry, how we should evolve our practices, and how we can expand and introduce potency and novel potency in our extracts and this will allow us to reach higher levels, which will allow us to get probably tapped into more of the biopharmaceutical industry, right? Because when you start to think about the concentrations of medicinal compounds and those compounds and drugs that are used in therapeutics, there's probably like a threshold. And typically with mushrooms, like you're thinking of taking this kind of stuff over a long time. Otherwise, you'd have to take a lot of mushrooms. You'd have to consume a lot, and it's kind of like impractical. But if we can reach new ranges, new ceilings, new tiers of concentrations of these novel compounds and bioactive compounds, we can create a new class of research and medicinal grade products. Have them certified. It's justified by our practice. This is like what I was talking about in the video, mycochemistry. It's like it goes beyond organic certification. You know what I mean? We have research-backed practices that draw the line, that create the mountain difference of potency and why you should go with this standard, and really not to just separate ourselves from other competition, but to provide additional options for those people that actually need this medicine. You know what I mean? A more natural option. So to not even keep it about competition and business like that. The whole point is medicine, right? So provide people more options for different medicines, therapies. Targeted isolation yielded 3 new sesquiterpenes. Yeah. So we have, what else do we have? Yeah, they use MS and NMR. Electronic circular dichroism analysis. I'm not familiar with that, y'all. I'm not. But I guess I could look it up. I guess I could look that up. Circular dichroism analysis. Okay. Oh, it's a spectroscopy, essential analytical technique used to analyze chirality in molecules. What? Through their optical activity. You're kidding. Wait, what? Why haven't I learned about this? What the heck? You're joking. I mean, we did like polarimetry. I learned about polarimetry and that kind of stuff, but I didn't use this. I didn't use that technique. That's kind of crazy. I mean, that makes sense, right? Again, what if we find a biosynthetic pathway that creates another configuration? What's the word, dude? Stereochemistry? Gosh, it's been too long since I've been looking at chiral molecules. Sorry, y'all. I'm pretty sure E&Z isomerization, right? We could be getting the enantiomers. Oh my goodness, enantiomers. Wowzers, y'all. That was a freaking brain fart for sure. Oh my goodness, enantiomers. There's like Nobel Prizes given out for the creation of certain molecules, and if we can find pathways in cultivation practices that can result in the creation of these compounds, oh my gosh, that's like someone's Nobel Prize waiting to be made right there. If that's you watching, you better give me credit. You better at least like some inspiration. Be like, yo, I saw this guy on YouTube and he was talking about this stuff. Y'all should go like, subscribe, and follow to him. Yo, but seriously, like in all honesty, like that's the potential that is here. That's the the Pandora's box we just opened with this, because you can vary the different strains you're using, right? It adds a whole new layer of foraging, right? You're already introducing new cultures, but new dynamics of those strains. Oh my goodness. I gotta take a second to just like take this all in, y'all. This is crazy. This is crazy. We just unlocked it. We just unlocked a whole new level of practice. It's adding variability. That's what we're doing. We're adding variability. We're increasing the ceiling for our potential to get these bioactive compounds. And once we start introducing higher level analytical testing, like this electronic circular dichroism, to identify the types of analogs we're getting in enantiomer structures and isomers, like, yo, we can map these pathways and utilize them for drug production. So so really there's so much room. Where is the room right now? If we're going to talk about this, where's the room for progress? We're capped in our in vitro cultivation, right? The nutrients that you provide are all it's got. It's one species, one immune system. So all the metabolic stress and work is done by that organism. Meanwhile, in nature, you got multiple organisms. You're not always competing, you're talking, you're sharing nutrients. You got some species that are tapped into a variety of conversations and interactions, dynamics, and species can play on those relationships. It's like when you network with someone who's really good at networking already, like you just tapped into a huge network. Meanwhile, in vitro, we're stuck in this box, which caps our medicinal potency. We are adding, we're breaking down the ceiling. We're blowing holes through the ceiling right now with our potential now. I'm so excited. And then when we look to nature, we can use that as clues as to what we should be co-culturing with. You know what I mean? Let's take it from foraging. They're in the wild. Mushroom has beneficial compounds. We find it. All right. We're thinking in co-culturing because we're thinking about having this species, which we're going to test, see its potency, see what compounds are being created. Compare that to us culturing it in vitro as is, right? Or if we can, or getting it to a generation as well that can finally fruit in vitro. Domestication. At the same time of foraging, we're going to get soil samples, bacterial samples on the wood, right? And we'll be able to isolate all the organisms that are around in that interaction where we found the fungi. That is a way for us to know what type of communication or competition is going on where you found that mushroom, to reach those levels of potencies of biochemical compounds seen when testing that strain. We can use those same species to co-culture in vitro, in the lab. Test that, see if we surpassed our level without co-culturing, right? Our axenic monoculture cultivation, and then compare it also to the wild. And who knows what we'll see at that point, because when you cultivate in vitro, look at the yields you get. We may see, just like you see a lot of fruiting bodies and really nice fruit, we may see a lot of the medicinal compounds when we intentfully introduce these other organisms, whether competitively or non-competitively, at different concentrations and at different stages of cultivation. That's where the variability, the research comes. It's the hard part, but it's the fun part as well, because we have a proven logic. We have an idea of where we need to be getting to. And then that's where everyone's art comes in, your own scientific art with all of this.